Welcome you all. Myself, Mr. S.S. Mahamuni, Assistant Professor from the Department of the Pharmaceutical Chemistry, Satara College of Pharmacy, Satara, affiliated to Dr. Babasaheb Ambedkar Technology University, Lunere. Today we are going to see the practical of the subject instrumental method of analysis, that is exam number 13. Nothing but separation of plant pigment by column chromatography. So we will start with the first, that is the aim. Aim is that to separate, isolate and identify given sample of mixture by the column chromatography. Then we'll move towards the objective. The objective of the experiment is first to understand the experimental technique for adsorption column chromatography. Second is to study qualitative aspect of column chromatography. Third is to study how to separate, isolate and identify given sample by the column chromatography. Next point is the principle. So the column chromatography is based on the principle of the adsorption coefficient as well as partition coefficient. So it is depending upon what type of the stationary phase we have to use for the column chromatography. So it may be partition coefficient based or it may be the adsorption coefficient based. So in the adsorption column chromatography, a mixture to be separated is dissolved in a suitable solvent and allowed to pass through the tube containing the adsorbent. The component which has a greater adsorbing power is adsorbed in the upper part of the column. The next component is migrated to a lower portion of the column and this process is continued. As a result, the components are separated and adsorbed in a various part of the column in the form of the different band. So in the portion of the column. Then next is the requirement. So first is chemicals. So it required silica gel for the column, then chloroform and methanol. So specific particle size silica gel column is used for this column chromatography. Next is the glassware requirement. So pipette, beaker, then measuring cylinder, funnel, etc. we can use for this experiment. Next is the apparatus. So this is a important apparatus, that is the column, that is a glass column, that or it is called as a chromatographic glass column, so which is having the specific characteristic features, that is it have the knob, it is having the filter. If it is not have the filter, then we can use the cotton to avoid the passing of this slurry or the stationary phase through this column. Then this knob is used as an actuator to actuate the release of the mobile phase through this column. Then we'll move toward the next stage is procedure. So first of all, this is the practical based upon the separation of the different types of the component from the natural origin. So that we want to use the plant material. For example, spinach leaves we can take for that, and we triturate that particular leaves of the spinach and used as a sample. So at the first, suitable solvent was selected for the given mixture. So it becomes the mixture of the different types of the pigments which is present in that spinach sample. Then silica gel for column was taken in a dish and activated by drying. So we dry or activate the silica by using the hot air oven. That is specific period of time for 30 minutes, 120 degrees centigrade. We set all these activation process of the silica gel. Then we use the silica gel for the column. Then slurry of the silica gel was prepared by using the chloroform. So specific solvent we use or specific type of mobile phase also we use to prepare the slurry of the silica gel. There are two types of methods are available to packing for the packing of the column, that is dry packing or wet packing. Dry packing, that is simply we put the dry powder and then we add the solvent or the mobile phase and pack the column, and wet packing is that we first of all make the slurry and pour into that column. We can use any type of the method as per your convenience. So then now one column take and cotton was fixed at the bottom of the column. If it is not having a filter at the bottom of that column, we can use the cotton and fixed at the bottom, and then slurry was poured into the column and allowed to settle down. So it become packed properly. Then sample is required quantity, that is sample of your spinach leaves extract is in a suitable solvent was added into the column. So slowly we add the sample at the top of the column. Then when sample solution absorb in a adsorbent, mobile phase was added. Then we add the mobile phase continuously. Then mobile phase added after the time interval so as to separate the sample. So after continuous addition of the mobile phase, there is a separation of different bands that takes place. So two samples were isolated. So we can also isolate two sample. Most of sample means in this case the different types of bands of the color pigments are separated. Most of 3-4 bands are formed. So it is depending upon what type of the mobile phase we have to use for this separation of this component. Then identify each sample with, or each instead of sample the pigment with the help of a TLC by comparing with the standard. So we take a different type of a standard color component and we compare with our separated component, that is beta carotene, chlorophyll pigments and all. So this is the diagram that will give the information about how to load the sample, how to prepare the column, and how to separate the component, how to get eluted component as in by this figure. So this is the preparation of column, then addition of mobile phase and sample, separation of component, and illusion of the one by one component. So these are the references we have to use for this experiment. So thank you all of you. I think you all understand this process of the separation of the component by the column chromatography. Thank you all of you.